Welcome, new MBI students. My name is Alvaro Sagasti. I'm a professor in the MCDB department. Although this may be an awkward time to be starting graduate school, I think it's actually a really exciting time to be a cell and molecular biologist. And that's because of the revolutionary new tools that we have for microscopy, which allow us to see details of cellular organization we didn't think was possible even a few years ago, and CRISPR-based genome manipulation, which allows powerful methods to study gene function. So my lab is taking advantage of these cutting edge tools to address fundamental questions in cell biology. So the broad question that motivates us is how cells adopt their shapes. So you know that cells are not simple spheres or cubes, but they are highly irregular and bumpy. Adopting these elaborate shapes is important for their function. So what we know about how cell protrusions form generally is that they are created by cytoskeletal filaments that shape the membrane from the inside, similar to how tent poles shape a canvas. And it's the specific arrangement, stability, and properties of cytoskeletal filaments that determine the properties of the protrusion. So our goal is to understand how cytoskeletal filaments are regulated to create these protrusions. So my lab studies both neurons and epithelial cells, but I'm going to tell you about an epithelial cell project that is at an exciting point. So specifically, we are studying beautiful, highly organized protrusions called microridges, which are found on mucosal tissues like your cornea or the inside of your mouth. And these protrusions are thought to help retain a protective mucus coating on those surfaces. We study these protrusions because very little is known about them. And so by studying them, we could discover new principles of cell shape determination. So specifically, we study these protrusions on the skin of larval zebrafish. And so fish, like your cornea, are covered in mucus, and therefore their skin cells are covered in microridges. But because zebrafish develop outside their mother, and we have a lot of genetic tools to manipulate them, they're ideal for studying these structures. So what you're seeing here is an electron micrograph showing the fish skin. So each of these polygons is the surface of a cell. And you can see in this close-up that these elongated microridge-like structures are arranged in these amazing maze-like patterns. So we knew that microridges are filled with actin filaments. So we realized a couple of years ago that we could study them in live animals by tagging an actin binding protein with a fluorescent protein and expressing them in those cells. And that allows us to make movies like this and watch the process actually happen in the live animal. And from these movies, we've learned a lot of things. But for example, with this movie, we discovered that microridges don't form as a single unit all at once, but they form from these precursors, which look like little puncta in this movie, that sort of coalesce to form and elongate the ridge. And this was a surprise because no other protrusion that has been studied is known to form from precursors like this. So our general approach is to identify cytoskeletal binding proteins that may regulate these microridges. And I don't have time to tell you about them today, but we've used RNAseq to determine the transcriptome of these cells, and then identified candidates that bind to actin, tag those candidates with fluorescent proteins, and then express them in live fish. And in this slide, you're just seeing 6 of about 20 proteins with which we've done this. And that has allowed us to identify several exciting candidates that localize to these ridges in really intriguing patterns. So once we have these candidates and we want to understand their function, then we can go in with genetic techniques. For example, creating CRISPR deletions of these genes that create mutant zebrafish, and then look at these microridges. And for example, envoplakin and paraplakin, these super interesting proteins that I wish I had more time to tell you about today, are absolutely required for this early process of microridge precursor coalescence. We can also use pharmacology to temporally control protein function. So for example, this inhibitor of the motor protein myosin, if we add it on early, we also prevent microridges from forming, telling us that the force-creating activity of myosin is required for microridge formation. But if we inhibit myosin activity late, we find that microbridges form normally, but they don't get arranged into these really beautiful, well-spaced maze-like patterns, telling us that myosin activity acts at a later step to organize and arrange these microbridges. So it's through experiments like this that we've been able to break down this morphogenetic process into multiple steps. And we started to just know a little bit about some of the molecules that are involved in these steps. But there's still so much more to know. So I can think of many exciting rotation projects. So if you're interested in this project, please email me and I'll be happy to talk to you about them. The lab currently is on the small side, but we are in expansion mode. So I would be very excited to attract new students to take on this project. So thanks for watching this video. Have fun, and I look forward to meeting you soon.